In this final part of today's lecture, and indeed the final lecture for this semester of Genes, Genomics and Human Health, we will consider the question of whether genome-wide association studies have contributed towards personalised medicine. In other words, have they been translated to the clinic? So genome-wide association studies, as we know, they provide an unbiased approach for the discovery of mechanisms and pathways that underlie human characteristics, diseases, and drug responses. We also know that these common illnesses that we've looked at this semester, type 1 diabetes, type 2 diabetes, schizophrenia, Alzheimer's disease, or late onset Alzheimer's disease, all of these type of illnesses, they contain many loci, they are caused by many loci across the genome. As a result, it's very difficult to identify single gene loci that impact on therapy. If you think back to the previous lecture where we were able to identify EZH2 and SMARCA4 as targets for improving therapy of lung cancers, that's certainly not the case of these multi-locus polygenic diseases that we've been talking about. What this slide shows is it just puts that in perspective a bit. So this is a breakdown of all the studies done in genome-wide association studies. We can see that there's a total of 2,457 studies, and this is based on a figure from 2016, of which well over 1,000 or so studies were done on specific diseases. Here, for example, asthma, diabetes, and so on. Just under 1,000 studies were done on traits, so this would be height, blood pressure, and things like that, whereas the remainder, under 10% of studies, were pharmacogenomics. In other words, can we map the link between genetics and response to therapy? And that's really shown here as well, that we can see pharmacogenomics, the number of studies of GWAS that look at this interaction between therapy and outcomes is very low, compared to the very high number and continuing high number of GWAS. What's interesting though is if you look at this curve, you can see that really in the past 5 to 8 years, there has been an acceleration of GWAS, but it looks like now if you follow this through, you could almost extrapolate that GWAS are starting to taper off a little bit. I think this tapering off is something that I've talked about before this semester, which is that GWAS are very, very good at mapping common alleles. And we're just about there with doing that. And we're now moving on to functional studies and also using next generation DNA sequencing to map rare variants as well. So perhaps in the next 5 years, the landscape of how we approach genomics will change significantly. What's also important is when we look at the ethnicity of populations that were studied in terms of pharmacogenomics. And overwhelmingly, the studies have been done on European populations, 78.2%, whereas other populations very much make up the minority. And you can see that here as well, the type of diseases that were studied. By far and away, the greatest number was cancer. Almost 50% of all studies of GWAS were done on cancer, followed by psychiatric illnesses such as schizophrenia, cardiovascular, asthma, a long way behind, and so on. So really, you could argue there's very much a biased approach in these GWAS, overwhelmingly European, overwhelmingly disease-based, and very small numbers of other cohorts or populations, and very few studies on the pharmacogenomics of genetics. Now, I don't think this reflects any conscious bias. It probably really just reflects where most of the biomedical research is done in the USA, Britain, Australia, these Western countries, which would focus they would get their cohorts mostly from European type ancestry. And the second part, of course, is that the first wave of GWAS was all about understanding heritability, understanding the genetic basis of disease. I think the expectation is that if we were to revisit these two graphs that we're looking at now in 10 years' time, that we'd see much more of a normalization, and we would see increasing numbers of other ethnicities, and also other diseases being looked at in the context of drug genetic interactions apart from cancer. So what we're going to talk about today is something called comorbidity. This is based on the understanding that patients who have chronic illnesses don't show just one illness, but in many cases show multiple illnesses. And what that tells us is that different illnesses are actually not that different, but they have shared genetic loci. Realizing this and mapping those pathways that overlap in different diseases provide an important first step in translating the findings of GWAS to the clinic, and we'll see this in the next few slides. So as we know, the goal of GWAS is to provide an understanding of the genetic determinants of common illnesses in populations. The small effect sizes of these variants meant that our sample sizes had to be very large. And of course, GWAS rely first and foremost to access from clinical health records of many individuals. We're first of all going to start with a remarkable study done in Denmark, which is not a GWAS, but it starts to present really important data on comorbidity of disease. Now, if you look at the title here, it says temporal disease trajectories condensed from population-wide registry data covering 6.2 million patients. So in other words, what they're looking for is the development of disease over time in the population of an entire country. And in this case, what you can see is they've looked at the electronic health registry covering the whole population of Denmark. And what they wanted to know is that they said, if we're going to be better at delivering medical outcomes, we need to understand how disease progresses. We need to understand that if you present, for example, with type 2 diabetes, what are the most likely complications? What other diseases are likely to start occurring if you have type 2 diabetes, and so on. So this study examined the entire spectrum of diseases and over a 14.9-year period of 6.2 million people, and it looked at over 1,000 different disease trajectories. In other words, it looked at the development of diseases in many different circumstances. I think it's really important, before we go any further, however, to really emphasize that this material isn't available to everyone. It's covered by very strict privacy protection acts, and it can only be accessed following application. Now, not everyone is going to be successful. As a matter of fact, it would be very difficult to access this data, and it would be protected by rigorous privacy legislation, as well as the absolute requirement to submit an ethics application, which would include in it rock solid guarantees on the protection of privacy of all individuals who are being studied. And so this is something that appeared in the paper to show also the references for the ethics applications and so on, and the rules under which this research was conducted. So what did they find? This is an example of their analysis. Now, incredibly complex looking diagram. What we need to do is look very carefully at what it's saying, and there's no way we're going to look at all of this diagram. So first of all, let's see what we've got. We're looking at the number of patients. The thickness of the line reflects the number of patients. The thicker the line, the higher the number of patients. So wherever you see a very thick line like this, we know we're looking at a connection that is relevant to at least 100,000 individuals. Let's look then at non-insulin dependent diabetes. That's here. Interestingly, there's a group that if they have asthma, they will progress to type 2 diabetes that we see here. Now, this is diabetes, and of course there are a number of ways that this can develop. By far the most common is this first arrow here, which is a multi-disease phenotype. For example, this is diabetes that we lead here, and then it can branch off into renal failure, ulcers, and so on. But what they found is a very important diagnostic feature is retinal disorders. In other words, if you have type 2 diabetes and you have your eyes tested and they find that there are changes in your eye, such as lipid deposition and so on, that this is actually a predictor of vascular disease. So by viewing these very large population studies, what they found is that diabetes is associated with changes in the eye, which is due to changes in lipid deposition, and that then leads to more systemic changes in the vasculature associated with changes in lipid deposition and function. So type 2 diabetes is just the first part in a much more widespread and different types of diseases that afflict these individuals. So it's showing us that you don't just have type 2 diabetes, but at some point in your life, you will start to develop retinal disorders, and these retinal disorders will occur at the same time as vascular disease as well. This is a temporal projection of a disease. Now, of course, not everyone will follow that trajectory. There will be some people who do not develop the retinal disorders, and there will be some who do not develop this widespread vascular disease. So already what we're seeing is we're seeing for a significant number, there are type 2 diabetes and other significant health deficits. But for other people, of course, they will just have type 2 diabetes in a less severe phenotype. And the understanding for that is, of course, it's probably partly environment, but it's also genetics. So when we see these incredibly complex diagrams and the various ways that disease can develop and the fact that people can have multiple chronic diseases, what we're looking at is not just the environment, but we're looking at differences in genetics between these groups of people. And that really raises the question is, to what extent are these disease trajectories genetic? In other words, to what extent does the development of one disease lead to another, lead to another, lead to another? To what extent is that genetic? Secondly, given that a disease such as diabetes has a number of comorbidities, for example, diabetes and vascular disease, is there a shared genetic basis between these different phenotypes, vascular disease and type 2 diabetes? And can that be used in the context of therapy? So this is now starting to get into the realm of asking clinically relevant questions of GWAS. And we're going to look at a paper. Its title is Analysis of 5 Chronic Inflammatory Diseases and 27 New Associations, these are genetic associations, that highlights disease-specific patterns at shared loci. So what's that saying? It's taking 5 diseases, it's saying that they have a lot of genetics in common, that's their shared loci. So GWAS have shown the overlap between genetic susceptibility to human diseases. Think back to our lectures on immunology, and in there, that there were many inflammatory diseases which showed significant overlap in terms of the alleles and the genes that contributed to those diseases. These genes in many cases were what we call pleiotropic, that they were expressed in and they had an influence on different cell types and different organ types. The diseases this paper looked at were ankylosing spondylitis, Crohn's disease, psoriasis, primary sclerosing cholangitis, and ulcerative colitis, which I'll come to in a minute. It's based on the study which I've just described done in Denmark. The study in Denmark raised this very real possibility that many individuals in a population have comorbidity of different chronic diseases. So instead of looking at vascular disease as a phenotype and diabetes as a phenotype, we're now looking at someone who has type 2 diabetes as someone who also has vascular disease. And what the authors do is they use this term of hidden molecular taxonomy. In other words, we start classifying disease differently. That if you have a chronic disease, we also need to consider whether you are defined by several chronic diseases. So cross-disease genetic studies provide an opportunity to resolve overlapping associations and look for shared pathologies. So in other words, genetics is giving us a window into the molecular basis of why someone might have type 2 diabetes and retinal problems and vascular disease. So just briefly, ankylosing spondylitis, this is arthritis that primarily affects the spine. Crohn's disease is an inflammatory bowel disease, the lining of the digestive tract. Psoriasis, inflammatory skin disease. Sclerosing cholangitis, it's in which the bile ducts in the liver become inflamed. And ulcerative colitis, it's ulceration and inflammation of the colon and rectum. So at first glance, some very different diseases, but of course they share an underlying inflammatory response. So the goals of this study were fivefold. They wanted to identify subsets of 5 phenotypes with shared genetic loci, and they did this by comparing genome-wide studies from each disease and also people showing multiple phenotypes. They wanted to identify additional loci. In other words, they're still on the hunt for this missing heritability. They wanted to investigate comorbidity and pleiotropy. In other words, if you have multiple diseases and if you have the same gene and variants popping up in different diseases, it would suggest that gene is pleiotropic. In other words, it has effects on different cells and different organs. And finally, they wanted to apply that to understand shared pathways and biological mechanisms common to these phenotypes that they were studying. And with that, I think we can say that we're getting closer to the clinic. In other words, if you have all these comorbidities, if you have more than one illness, can we approach that as a single illness? So if you have type 2 diabetes and vascular disease, instead of seeing that as distinct, let's find the commonalities of those 2 diseases, and perhaps there is a single way of treating those symptoms. It's probably a simplification, but that is in an ideal sense what they were looking for. What we see here is a summary of their genome-wide association studies. Each little dot in the color represents each of the 5 diseases, black, blue, pink, etc. And what we can see is there are some loci, for example, here contains associations with 4 of those illnesses, whereas others, for example, here is a locus which contains a variant across all 5. And you can see that there are many instances across the genome that contain associations with 3, 4, or 5 of these different illnesses, clearly indicating a shared genetic susceptibility. Now, the outcome of that, of course, if you imagine what we've just said is that we had 5 diseases, and in many loci across the genome, there is a gene and a variant or variants in that gene, which increases susceptibility to perhaps 3, 4, or 5 of these diseases. So what they've done is they've said, okay, let's look at the genes which show the highest association with as many of these 5 diseases as we can find. And this is what's called a protein-protein interaction network. If you imagine a cell is full of pathways where one protein talks to another, whether it's through a kinase cascade, whether it's in the nucleus where transcription factors are interacting with other transcription factors, proteins talk to each other. And this diagram here is not for us to understand and dissect, but it's simply to illustrate that there are networks of protein interactions, whether they are direct physical interactions or functional interactions. And where we see these green boxes, these are some of the genes, JAK2, the interleukin-2 receptor, etc. These genes in green boxes have come up across 2 or more different disease phenotypes. And the reason that these are highlighted is because of this diagram here. What it's showing is, here are the names of genes down in red. These genes contain alleles which are associated with 2, 3, 4, or 5 of the 5 diseases that we've been talking about. In blue is a drug which targets this gene. For example, the protein JAK2 is targeted by a drug called tofacitinib, and that can treat psoriasis and ulcerative colitis. NF-kappa B1, for example, can be targeted by 3 different drugs, and from these 3 different drugs, there are a range of these: ulcerative colitis, psoriasis, Crohn's, and ankylosing spondylitis, for example. So what they're saying is we'll do the GWAS, of these 5 diseases. We'll look for shared genetic loci between these diseases. We'll identify proteins at these shared loci that talk to other proteins. And we'll identify drugs that can block, modify these protein interaction networks. In this way, we can start targeting these phenotypes where someone who has, for example, psoriasis may also have ulcerative colitis, and now we can think about treating their different symptoms in a much more targeted way. So really what this paper is all about, it's saying if we start thinking about chronic disease as not just a single disease, but as coexisting comorbidities. In other words, you have not just psoriasis, but you may also have ulcerative colitis. We don't treat them separately, but we start investigating ways of treating them as a single related disease. And that's really what this GWAS paper is doing. So what they've shown in this study is that there's a presence of shared pathological pathways and to explain the co-occurrence or comorbidity of 2 or more of these 5 illnesses. Patients with showing these multiple phenotypes are genetically distinct from patients who may just have psoriasis, for example. So in other words, there is clearly a genetic basis for individuals who show psoriasis plus colitis plus perhaps spondylitis, for example. They also know that although they need to do more investigations, they propose that target gene-drug combinations selected by this approach could represent promising candidates for novel drug discovery for the treatment of these different inflammatory conditions. So really what I've done here is I hope I've selected an example where we use GWAS not to think about a single locus and a single disease, but to use GWAS in the context of understanding large collections of health records to really start rethinking how different diseases are related to each other. And given that we're talking about common illnesses in a population, many of the diseases we're talking about are chronic. So that if you have type 2 diabetes, you may also have vascular problems. If you have psoriasis, you may also have ulcerative colitis. Not always, not for everyone, but it may happen. And if these diseases have overlapping phenotypes, then that may provide a new way of treating these very complex, chronic and at times common illnesses.